So, welcome to the American Brain Coalition's 2024 Educational Series webinar, Open Science Drives Discovery. I'm Dr. Bill Greiser, teaching assistant professor in psychology and interdisciplinary neuroscience at Portland State University, the neuroscience coordinator and co-founder of an outreach nonprofit called Northwest Noggin, and a proud member of the American Brain Coalition. I'm also a programming, engagement, and outreach committee member for the ABC. I'm very excited to moderate today's educational webinar on the transformative power of open science. This session will delve into what open science means, like what is open science, the importance of sharing data and which data, and how it fuels scientific innovation. We're going to learn a lot of valuable insights on how open science is shaping the future of discovery and how you can actually be a part of this movement too. So we're going to be lucky today to hear from two expert speakers, and we're going to allow the last 15 minutes or so for a Q&A with our speakers. Please feel free to ask questions throughout this presentation in the Q&A box. So you should see that Q&A box there. Please place your questions right in there, and then we promise to get to them. But for now, let's just jump right in. So we're going to hear first from Benjamin Dichter, PhD, the founder of Catalyst Neuro and community liaison for Neurodata Without Borders. Dr. Dichter received his PhD in bioengineering from the UC Berkeley UCSF Joint Program in Bioengineering in Dr. Edward Cheng's lab. There he used electrocorticography, ECOG, to study the neural control of speech in humans. Much of this work focused on how we control the pitch of our voice when we speak and when we sing, right? He is now a data scientist consultant for neuroscience labs, focusing on building systems for the sharing of data and analyses. Dr. Dichter?
Thanks so much, Bill. All right, I'll share my screen here. Hi, everyone. Again, I'm Ben Dichter, the founder of Catalyst Neuro and the community liaison for the NWB project. Catalyst Neuro is a team of research software engineers that specialize in connecting data producers, tool builders, and data users in neurophysiology research. I'm going to go more into detail about what that exactly means. But first, I just want to introduce, the types of problems that we're solving here. So neuroscience research, it's used to study the nervous system, how the nervous system works, especially the brain. It studies, the normal functioning, and it also is used to study diseases such as Alzheimer's and Parkinson's. And there are lots of different ways to do neuroscience research. Here, I'm showing a plot here on the left that shows, a bunch of different types of recording modalities that span different temporal resolutions and different spatial resolutions. So to answer different types of questions, we need to use different types of technologies. And on top of that, there's, the things produced by neuroscience research. It's not just the data, but also the analysis software and metadata, the data derived from those analyses. All of these are research products that we want to facilitate sharing to accelerate science and to improve the quality of science. So we need, there's a huge need in neuroscience for open data, primarily because the data that we collect is very expensive. So it's expensive in just in terms of money. That most of this research is funded by taxpayer money, by government programs. And so it's important for us to make sure that this is a public resource that everyone benefits from. It's also expensive in terms of time, and that's in two different ways. One is these experiments can take years to go from conception to publication, and that's a valuable time where people are suffering from neurological diseases, and we want to accelerate that. It also takes a lot of individual time as a resource. I mean, it takes years from very highly trained people to do these experiments and to do these analyses. So time is an enormous resource that we want to use efficiently. And then thirdly, a lot of these experiments that we do use animal lives, and they often involve sacrificing animals. So it's really important that we derive as much value from this data as we possibly can. And we know that often when you collect data for one scientific question, it can be used to answer many other scientific questions. It's true all the time. But sharing neurophysiology data and neuroscience data in general with your collaborators can be very tedious. There are just dozens of different, diverse, complex formats. If you record data from one acquisition system, it's not clear that your colleague will be able to read it if they're used to another acquisition system. These data sets are very high volume. Often we're talking about terabytes of data. Physically, how do you even share that with someone? The metadata is often incomplete. You have kind of knowledge within your lab that that you know how to read the data, but someone else trying to read it might not be able to understand. Like, oh, what was the sampling rate here? Like, and that type of information can be lost if someone goes to another lab and they're not reachable. And so then all of that data, all of that data could be made useless by not including a very simple piece of metadata. On top of that, it's really hard to share scientific software with colleagues, software for data processing and analysis and visualization, because that software is often built on top of very specific data formats. And so it requires converting data from one format to another, which sometimes isn't even possible. And that makes it really difficult to share scientific software as well. As a result, without the necessary tools to share data and to share software, it's often easier for labs just to collect new data and build new software. Even though that takes years, they can trust it more because they know they're not going to run into this problem of having problems with the data and the metadata. And we think this is enormous waste. So we envision a future. I think it's people who are really passionate about open science. We envision a future where a neuroscientist, instead of when they have a scientific question, instead of thinking, what experiment should I design to answer this question, they think, what query can I run on existing data to download existing data that could be relevant to this question and analyze that instead. And we think this could rapidly accelerate scientific progress if this were easy to do and increase the quality of science. So we've been working for years over how can we build the infrastructure to enable this other way of doing science that's much more collaborative. So luckily, there has been this new program by the NIH called the Brain Initiative, started back in 2014. And this Brain Initiative, for the first time, has really dedicated the right amount of support to these infrastructure issues that facilitate open science. So there's an informatics component to this Brain Initiative that has three branches. So one is to create data standards, common languages for describing data and metadata, so that labs can easily communicate with each other through these data standards. Data archives, services where data can be shared. So literally, how do you upload and download this data set and search across all of these data sets? And then open software for analysis and visualization of this data that's integrated with the standards and the archives that creates an entire ecosystem of not just standardization, but also facilitating the process of scientific discovery with this open data. And recently, the NIH has now created a policy where they require data to be open in neuroscience. So starting in the beginning of 2023, shared scientific data should be made accessible as soon as possible and no later than the time of the associated publication or at the end of the grant performance period, whichever comes first. So if you publish a paper, you have to publish the data alongside it. And if you have a grant and you don't publish, then you have to publish that data anyway at the end of the grant. In addition to this, proposals must have a data management and sharing plan where they outline how exactly they're going to comply with this standard. So when we think about open data, there's been this rubric that's was established in a paper in 2016 called FAIR. So FAIR stands for findable, accessible, interoperable, and reusable. So the open science community has really rallied around this as a rubric for how to assess data that's shared, because simply just throwing data up and making it available isn't necessarily going to be useful if it isn't findable, right? If someone who wants to use that data isn't able to identify it as something, as a data set that would be useful for them. So that's one thing. Accessibility. Okay, once they find it, can they download it? Can they access it? Can they actually integrate it with their science? Is the workflow for them actually getting a hold of that data reasonable? Interoperability. So do you provide the necessary tools so that data analysis and visualization tools can integrate with this? Is this data connected to other data sets in a way that allows you to aggregate or compare between different data sets? And ultimately, is it reusable? Is the data of high enough quality that somebody could take it and use it and derive new scientific insights from this data? So I want to narrow in a bit on neurophysiology data specifically, because this is the area that my company and I have been involved with. So neurophysiology is information recorded from the functioning nervous system, particularly the brain. It's used to study functions like sensory processing, so vision or hearing. It's used to study behavior and cognition, like decision making. And it's often used to study diseases like Alzheimer's and Parkinson's, and often to study, for instance, sensory processing during, that's associated with the diseases of, for instance, Alzheimer's and Parkinson's, or like the motor control and behavior, for instance. So the landscape of experiments is really quite diverse. So I'm just showing kind of a few examples here. There's a lot of work done in rodents. So these are mice and rats. Sometimes they're running on tracks and have rewards. Sometimes they're running through mazes. Also, it includes work done on people. So you might have a person doing like a speaking task. For instance, this is kind of a nod to my my prior work, as Bill mentioned. You could record neural activities using different methodologies. So you could use optical methods to look at neural activity. You can use electrical methods. But at the end of the day, you're going to end up with a time series. So neural activity over time. So here is what the results of like an optical experiment would look like. We're using a microscope to measure neural activity. Here's what it would look like if you're extracting action potentials from an electrical recording. And here's another type of electrical recording where you're looking across larger regions. But in each of these cases, you have time series of neural activity over time that you want to relate to some function or behavior or cognition or to some disease. So we want to share this data. So we've been working on an ecosystem of tools that enable you to do this. This has been work funded by the Brain Initiative. So the first is NWB, Neurodata Without Borders. So this is a standard for neurophysiology data and behavioral data that is a common language that allows one group to convert to this standard and then another group to be able to understand the data. And then there's the Dandy Archive, which is an archive and collaboration space for sharing and analyzing neurophysiology data. So once you convert your data to NWB, you can share it using the Dandy Archive. So I'm part of both of these teams. They are led by others, so I'm in collaboration with larger groups that are developing both of these resources. So first, Neurodata Without Borders. So it defines a unified data format for neurophysiology, and the goal is to package the data with all the metadata necessary for reanalysis. So of course, this includes those time series that I mentioned, the electrical and the optical recordings, but it also includes information about the data acquisition system. What was the sampling rate? What were the settings? What was the experiment design? Are there stimuli? Are there trials? Is there environmental changes? Information about the subject, such as the species and the age and the weight, the behavior of those subjects. And it also defines an extension, which allows you to, an extension framework. It allows you to build upon the existing standard so you can define new data formats that correspond to new acquisition systems or new analyses, because the field of neurophysiology is always evolving, and we need an ability to adapt to these changes without having to completely reinvent the data standard. So to convert to this data standard, we have recently released an application that we call NWB Guide, which stands for GUI for data entry, or graphical user interface for data entry. So this, you can think of it as kind of like a TurboTax for converting data to NWB. It allows you to convert data with minimal time and effort. It supports currently 45 different common data formats, and I'm sure as soon as this talk is published on YouTube, it will be more than that. It extracts the format specific metadata from each of those formats, and it automatically applies data engineering, which performs lossless compression on the data. So you're able to reduce the file size without actually changing the values of the data themselves, and at the same time optimizing for cloud compute. It can be done at the terabyte scale, so we can handle really large data sets from the latest technologies. And you can compose conversions together from multiple different data streams. And finally, then once you convert to NWB, you can actually upload to the Dandy archive. So in addition to making conversion to NWB really simple, we also want to allow you to access state-of-the-art tools once your data is in NWB. So we've collaborated with dozens of analysis and visualization tools, really state-of-the-art tools across different domains, including electrophysiology, optical physiology, such as calcium imaging, behavior, data management and publication tools, data exploration tools. These are tools that we don't develop ourselves, but we work with the community. We collaborate with them to make sure that these tools can read and write NWB files. If you want to learn more about this, we have a recent eLife paper that's publicly available. Speaking of open science, we love eLife as a component of the open science ecosystem. So feel free to just go to the eLife website and search Neurodata Without Borders. You'll find this paper. You can read a lot more about it. Okay, so now I'd like to talk about the Dandy archive. So this is an archive for publishing and sharing neurophysiology data, including electrophysiology, optophysiology, and behavioral time series, and images from immunostaining experiments. So Dandy stores neurophysiology data and associated behavioral data in the NWB format. That allows it to extract metadata from NWB and allow it to expose it for search, as well as validating NWB files. Once you convert data to NWB, you can actually upload hundreds of terabytes of data to the Dandy archive for free. So this is a really amazing resource. It can handle all the raw data from most neurophysiology experiments currently. And the way it's able to do that is there's actually a partnership between Dandy and Amazon. So in addition to being an online retailer, Amazon is a top cloud service provider. They provide cloud storage and cloud compute. And they have this program called the Open Data Sponsorship Program, where they allow scientists to post their data for free. So you can upload data for free and download data for free. So we pass that on to the neurophysiology community. And with the help of Amazon AWS, you can actually do all of this, hundreds of terabytes for free. So that's pretty cool. There are currently about 100,000 sessions of neurophysiology data on the archive across almost 300 individual data sets. This includes really high-profile data from like the Allen Institute, the International Brain Lab, HHMI Genelia, the Microns Project, if these ring a bell. They're really large-scale efforts to collect neurophysiology data, often across multiple labs. But they also, there's a lot of submissions from individual labs that do really high quality science and are able to share their data through here. We're really excited about this adoption curve where you see there's this steady growth and it's actually accelerating. So we're getting more and more data sets every month than the month before. And we're really excited to see this massive adoption. It's pretty diverse data covering lots of different species. A lot of mice and rat, as you might expect, but also a lot of other species, including worms and fish. And the size of the data sets ranges from tens of megabytes to tens of terabytes. There are some really high value data sets that are very small, but there's also really huge volumes that can be mined for all sorts of insights. Okay, so I would like to actually do a live demo really quickly. Here's a data set from the Moser Lab, which was attributed as part of their Nobel Prize in 2014. So we worked with the Moser Lab to get a hold of this data, which they were sharing openly, but we helped convert it to NWB and publish it on the Dandy archive. So now it is available. So I actually just going to quickly show you what this looks like. So I'm just going to go to the dandyarchive.org. And I'm just going to type Moser here in the search bar. This is the data set I want. And this is the Dandy set landing page. It has metadata that shows you information about the data set. I'm just going to dig into the actual files. You don't need a Dandy account to do this. Literally anyone can do this. Anyone watching this, you're welcome to try this out. I'm just going to go to a random session of data here and say open with NeuroSift. So this is an app that was built by Jeremy Maglin from the Flatiron Institute that allows you to interactively view NWB data on the Dandy archive. So this data is being streamed from the archive into my browser and being rendered. So I'm just going to say I want to view the electrical series, behavioral data, and a raster plot. And I'm going to view all 4 of these. Now, this is an interactive view of this actual experiment. So this is an experiment where there were electrodes recording from neural activity as the animal was exploring a square environment, which was their cage. So here's the movement, X versus Y, and then this is X and Y over time. This is the actual electrical signal that was recorded from the brain. And then down here we have each tick here is the time of an action potential from 4 different neurons that were identified from this electrical signal. And if I go here, I can like play this and you can see what it looked like to have the animal moving around in this cage. So this is a dashboard that's just streaming the data. Because it's an NWB, it knows exactly what type of data it is and how to render it. And because it's on Dandy, it can access this data really efficiently and pull it in real time into this dashboard. And what's really cool about this is this software was not built to look at this data set specifically. It's built to visualize NWB, and it can be used to explore any NWB file on the archive. Okay, so now let's go and think about is Dandy fair, Dandy and NWB. So is the data findable? Well, you just saw me search for the Moser data set, so at least there's some level of search there. And yeah, you know, if you have keywords for specific types of data you're looking for, you can just pop it in the search bar, and you're likely to find good results. So it has some level of findability. Accessibility, yeah, anyone can access and download data for free from the Dandy archive. Even you don't even need an account. You can just go and do it. Is it interoperable? So that tool, NeuroSift, was built by a third party, right? Not someone from the Dandy development team, but it was able to be built because of all of the APIs and all of the tooling that was exposed by the Dandy archive to say, okay, here are all the data sets, here's what's in them, here's how to download them. So it has that level of interoperability that facilitates tool development. And is it reusable? So this is really the key, right? All of this is all well and good, but we really want to see, okay, are people able to take data from the archive and create new scientific discoveries using this data, right? That's the whole point. And so we've started to track this. We've started to look at neuroscience papers that actually use data, NWB data from the Dandy archive. And so far we found about 30. I'm listing a few of them here. I just want to mention one of them that was really cool was this paper that that established a new methodology called Zebra, and it was published in Nature recently. And one of the figures from this paper, this is derived directly from an NWB data set on the Dandy archive. And I was talking to the authors of the paper about this, and they said, yeah, we actually didn't even need to really do a lot of debugging with the contributors of this data. We were able to download it, understand the structure, and just generate this figure and integrate it with the rest of the paper. So we were just really excited to hear that because that's exactly what we've been going for. And through looking at these papers that are analyzing data, we've realized there are lots of different ways that you could do it. So one is a showcase, right? So this is what the Zebra paper was doing. They built a machine learning algorithm and they wanted to demonstrate how useful it is. And you can download a bunch of different NWB data sets and show off your data. And that's a great use case. Another one is a pilot. Let's say you have an experiment you want to do. Why not search the archive, see if there's an existing data set that's similar, where you can run your analysis on that, and maybe that will help you refine your experiment and get you to the next step. Another one is a continuation. You know, if you read papers, a lot of the times in discussion sections, they have unanswered questions. They're like, oh, maybe this is true, maybe that is true. We didn't have time to look at it. There's a lot of like meat left on the bone a lot of the times with these papers, and it's a great idea to kind of just download this and see if you can start answering those questions. You can do a confirmative analysis. Let's say you've finished your project, you're about ready to submit. What if you go onto the archive, find a similar data set, and see if you can reproduce those findings in an open data set? That can enormously strengthen your paper and strengthen the trust in those findings, ensuring that it's not due to some kind of idiosyncratic difference in the way that you did your experiment. Another possibility is contrastive analysis. So let's say you have a finding and you want to say, well, this isn't true in other species or in another brain area or in another recording modality or for another disease state. So, accessing open data set gives you this variety of data where you can start to assess these sorts of claims. Also, you can do aggregative analysis. This is where you're taking data from many different groups and aggregating it together, gaining more statistical power so you can have more sensitivity and you can ask more specific questions. And finally, there's the educational purpose. Any educator can leverage this, and they can have their students do analyses on real data using the real data sharing ecosystem that they would really use as a neuroscientist. And that can just be really inspirational, I think, for students and a great educational opportunity. So we really encourage educators to use this resource as well. So there's this energy barrier to data standardization that we're starting to overcome, and there are a lot of different ways to address this. One is what I would say sticks, funding ultimatums from funding agencies. This is effective, but not if used as the only method. Maybe a better approach would be carrots, right? Making it even more attractive to enter the standardized state where you have access to state-of-the-art analysis and visualization tools. And another way to address this is with guidance, right? Documentation, tutorials, hackathons, working directly with labs and providing support, building and distributing open source tools that makes it easier to adopt these standards. So we've been doing these trainings for NWB and Dandy, including in-person and virtual workshops, user days where you learn how to convert data, developer days, where developers come together and they figure out how to get their tools to work together better. Neurodata Rehack has been going for the last 3 years. That's a school specifically for how to access existing data sets and analyze them and integrate it into your own science. There's tutorials and presentations at conferences such as CoSign and INCF Assembly, eBro. We're going to be at SFN shortly. We do roadshows. So we will actually come to your school, your research institute, and lead a workshop for students there on how to convert their data to NWB, publish on Dandy, and use those resources. We have a YouTube channel where we post all of our talks for people who can't attend in person. We have tons of tutorials for Python and MATLAB. We have tutorials for the guide application that I mentioned. We have a Dandy handbook, and we monitor Slack and GitHub and email. So we're happy to talk to you if you're interested in using NWB and Dandy. Neurodata Rehack specifically was again, this hackathon where you're deriving scientific insights from existing neurophysiology data through secondary analysis. We've held this event at the Allen Institute in conjunction with the eBro conference and at the HHMI Genelia Research Campus. We've gotten a lot of great support from different foundations as well. So Catalyst Neuro, again, it's a team of research software engineers. We specialize in open data and software and neuroscience. And we've worked with the Simons Foundation and the Michael J. Fox Foundation to support 19 and 14 different labs. So basically, these foundations will fund labs to do research, and they'll also work with our team to ensure that the data produced by that research can be shared with the world. The Kavli Foundation funded the development of the NWB guide, and they have funded Neurodata Rehack. HHMI Genelia has hosted many NWB events, including NWB user days and developer days and Neurodata Rehack. We really love working with these foundations, and they've been hugely supportive of the open science effort. There's a lot more to open science than NWB and Dandy. We're just kind of a small piece in a much larger picture. There are other data standards. There are other data archives. There are research labs that are sharing data and analyses. There are other research labs that are using these data and analyses. There's this really cool trend where labs are developing open source acquisition systems. So they're actually like publishing the design documents for how to build these systems. And you can build them with cheap parts like cell phone cameras. And so it's actually amazing the quality of data you can get now with like cell phone based technology. And of course, preprints and open access journals are a huge part of open science as well. So in summary, open data can accelerate and improve neuroscience research, enabling more rapid progress in understanding diseases like Alzheimer's and Parkinson's. Open science requires shared infrastructure such as data standards and archives. NWB and Dandy are working together to enable fair data in neurophysiology. And neuroscientists are actually starting to use these resources to generate new scientific insights. And the movement has been supported by the government as well as by foundations. So if you want help, if you're interested in this and you want to learn more, you want to work with us, again, Catalyst Neuro is this globally distributed team, and we're happy to work with any labs that are interested in adopting open practices. I just want to thank, again, the foundations and the Brain Initiative for the support. And with that, I will pass it on.
Thank you very much, Dr. Dicker, for a very engaging presentation. It produced a number of questions in the chat that I'm going to hold to the Q&A at the end of the discussion today, but I really appreciate your thoughtful insights. I'm kind of excited myself, to be quite honest, because I could use some of that data in classes that I teach with undergraduates at Portland State. So let's now turn it over to our next speaker, Susan Tappan, PhD, who is the head of community engagement at the Spark Data and Resource Center. Dr. Tappan is a developmental neuroscientist with a keen interest in FAIR and open science. She received her PhD in biopsychology in 2001 from the University of Virginia, and she went on to further her studies in neuroscience up here in the Pacific Northwest at the University of Washington. Sue is also a member of the Spark Data and Resource Center. Her passion is identifying practical solutions to biomedical analysis needs that balance the essential requirement of reproducibility, biological relevance, as well as person-hour effort, to make that balance there. So, Dr. Tappan?
Thank you very much, Bill. And Ben, it's such a pleasure to be hanging out and talking open science with you and everybody today. I am thrilled to be here, and I'm going to present some information about the Spark program, which in a lot of ways is a great companion to the Brain Initiative, and are looking at with respect to FAIR data and our interest in open science. And so with that, so Spark is, I like to think of it as a bit of a sister research program to the Brain Initiative, where the Brain Initiative is really dealing with the higher order components of the CNS. Spark, which takes its name from the NIH Common Fund that initiated it. It stands for Stimulating Peripheral Activity to Relieve Conditions. And so this research program was set up to build together a large consortia of researchers to create infrastructure, as well as do primary research about the multimodal components of neuromodulation in the periphery, the control of organ systems, so how your heart beats, how your lungs work, and how your stomach and digestive system are coordinated and working together, are great when they work well, but they're also the site of many disorders that can impact the human condition. And so understanding how these organs are controlled by the autonomic nervous system, and what opportunities exist for modulating that behavior becomes an area that can have some real defined benefits for the research community and the human population. And so in the first series of the research program, we focused on setting up, a unified platform that brings together and integrates all of this data through the common standards, similar to what Ben has already introduced. And we link it all together with this powerful knowledge graph so that we can incorporate all this information together and utilize it going across species, across modalities, and across scales, so that information that may be performed in one organ or looking at one specific organ could actually be of relevance and utility when it comes to another data type or another experimental application. And so from the perspective of open science, Spark is really keen on creating a mechanism where you can discover what is currently available and known about the neural control of organ systems, see what other sites of interaction might be, and use that information together to pick and choose and build new ways to generate hypotheses or provide a view of the data from your own lens. And so the opportunity to drive unique analyses and discovery that is across species, across modalities, so bringing together things like gene function as well as physiology or imaging data. What does it look like in a rat versus how does that same structure differ in its organization when it's in a disease state or in a different species, are all things that are really natural questions for us as we get more curious about what individual organs are doing. And so the goal of the Spark Data and Resource Center is to bring together all of this data into a multifunctional online hub so that we can coordinate, synthesize, and predict additional activities. And so the history of Spark, as I said, it was established by an NIH Common Fund program to promote the development of more effective and targeted neuromodulation therapies. And initially, it started with focusing on 5 organ systems: heart, digestive system, respiratory system, and bladder. And the idea here was to see as targets for neuromodulation, how can we create a greater compendium of information about the anatomy, the physiology, and mechanisms for intervention, and bring all that information together so that we can start to develop a therapeutic approach to treat diseases of the autonomic nervous system with fewer off-target effects. An off-target effect is when you have activity in an area you didn't expect, and that can produce some symptoms or other types of responses that are just not optimal for what you were planning for. And so it is important to be precise when it comes to things like this neuromodulation therapy. So a classical example would be a pacemaker. You want to be able to stimulate the heart without overdriving the heart so that you are not causing undue stress. The initial period of research has produced a lot of great information, and in this next phase of the research period, we're focusing on the vagus nerve in humans. And so the objective of this component is to bring together, which it began in late 2022 and comprises 3 main projects. SPARC-V is vagus nerve mapping and physiology. And so it includes the goal to create a multi-site clinical study of the effects of vagus nerve stimulation, as well as understanding how much variation there is from person to person in vagal anatomy. The vagus nerve derives its name from the Latin word which means wanderer, and the vagus nerve actually goes a vast number of places. And understanding exactly where it goes and how variable that is from person to person is really important when you're trying to properly devise a mechanism for intervention. And so in order to create these new transformative therapies, we really need to understand how much variation there actually is. And then another type of activity that is being supported by the SPARK program, funded by the National Institutes of Health, really gets at the topic of creating these open source neuromodulation technologies. So Ben mentioned briefly that, some of the new avenues in open science is to create tool sets that other people can build for themselves or use or riff on freely and openly. And in this case, these two applications look at creating devices, so stimulating and sensing devices for neuromodulation technologies, so that those people, those research teams who want to examine neuromodulation for a particular therapeutic endpoint can utilize these devices and optimize them for their particular activity. Lastly, the NeuroMod Prize is a new program wherein the NIH is incentivizing the development of neuromodulation technologies through a competitive approach. So thinking outside of the box allows us to approach things in a number of different ways. And the NIH Office of Strategic Coordination has really done a great job through SPARK and the BRAIN Initiative to really envision how we can bring researchers beyond the confines of their own labs and the collaborations with the people that they know, and expand even further to invite people to come together because of shared interests and opportunities for novel discovery. This brings together this power of convergence by bringing together molecular medicine, neuroscience, engineering, and computational science to develop these devices for the diagnosis and treatment of diseases. So as I mentioned, the NeuroMod Prize is an actual prize that invites specific teams to further develop their research. And then on the other hand, we also have open source development where the source code and the device designs are going to be made available on the SPARK program portal so that you can take advantage of them for your own particular use. Therapies in development that are targeted for neuromodulation in the periphery, treatment options for heart failure, hypertension, diabetes, gastroparesis, incontinence, but also what we might consider more CNS activities like depression, epilepsy, PTSD, Parkinson's. And so because the nerves in the periphery are very accessible, it's much easier to access these nerves in the body than it is deep in the head. You have an opportunity to effect some neuromodulation to bring change in the central system from stimulating the periphery. And so that's a really exciting opportunity here. As I said, we're interested in the vagus nerve in this next set of data that is coming. And I want to just give an indication of what that ultimately is going to result in terms of just the sheer amount of data that's going to be comprised. This is the largest experimental project devoted to the anatomy of human vagal systems to date, and involves a very careful dissection, both molecularly and anatomically and physiologically, to understand where the branches are, how they vary between humans, and what that ultimately means when you're thinking about creating a new device or personalized medicine opportunity going forward. And so what we expect to have available on the Spark portal and beyond as a result of this entire research program that is a multinational research endeavor is a very large data set that you can then port for simulation studies. You can use to devise new devices. You can take this data and create experimental hypotheses that you can test yourself. And so the power of open science becomes real as the data becomes accessible and available to you. The nice thing about this data is that it is being created and crafted across multiple labs, but with similar data standards, and an eye towards making it well understood by the people who generate it, as well as accessible both semantically and from the data perspective, so that you can really just dive right in and start exploring. And so the Spark platform is where all of this peripheral and integrative systems biology data is being housed. So we provide a mechanism for people to freely share their data. You can access it free of charge, just like the Dandy archive, like Ben was saying. And our data is available on AWS as well. So you could even bring that data from, select some stuff from Dandy as well as some stuff from Spark, and utilize it together. And so one of the goals of this presentation is really just to expose you to some of the things that you can now do and find, because a lot of data is coming online, and it's going to be very, very exciting. So Spark offers this integrative multi-species repository. We are a what's called a domain-specific repository. So we're interested in peripheral nervous system data. So anything that has to do with basically how organs function, how your body categorizes sensory and motor information, as well as sympathetic and parasympathetic information. It's scalable and sustainable, and we store and provide the raw data, primary data, derived data, protocols, and code. So what we aim to do is make this data something that you can just start exploring. You can come in and say, okay, well, I'd really like to know if there's any data about CGRP nociception in rats. You can just key that up, look for it, and maybe you'll find some data about the lungs, and maybe you'll see some great mapping data about the spinal cord, about how these circuits are built, and really drive you in many different opportunities and directions. From that starting point, you can then start to create these questions that allow you to really explore how all of this data comes together and what's similar and what's different. And we want to put this data in context and in place. And so that includes mapping that data to a schematic diagram, so that you can look at what we call a flat map to get an idea of where this data might be from. So maybe you're not quite sure what that structure is. You can see it on a map and be like, oh, okay, now I'm starting to get a feel for where it is. Additionally, you can map that data to what we call an organ scaffold. So over here on the right is a finite element model of a heart with neural fiber information that has been captured from an imaging experiment, and then it's fit to a heart. And this digital scaffold can be deformed to fit the data, but it can also deform to model how the heart beats from beat to beat. So you can start to see how things change as activity changes. So if you start layering on electrophysiology data alongside the structural data, you can start to get a picture of how form follows function and how function can impact changes in the structure and activity levels. And so that's sort of what Spark is really excited to bring when you've got this ability to bring data from many people, many research labs together. We want to bring this meaningful community along with us so that you can get more from the data regardless of your familiarity with it. And so in order to do that, we work with investigators to ensure that their data is published in a meaningful way, because we want to celebrate the information that they and time and expertise that they have exerted to do such careful science, and use that as a jumping off board to create new collaborations. And so this allows you to answer questions that are impossible to answer by looking at individual data sets or files. You need that context in order to know what you're looking at. And so Spark started off focusing on the autonomic nervous system. And as of today, we have over 300 data sets and computational and simulation models on the repository so far from over 500 contributors, as well as over 1,000 active simulation platform users. OSpark is our computational simulation platform, which is free to use. Anybody can request an account, and so you can bring data from the Spark platform right onto the OSpark online portal and start testing simulations and building your own analysis pipeline. 100% of these data sets and models are available for reuse. One thing I'd like to just highlight really quickly is I captured this screenshot this morning, and just the variety of data that is available on the Spark portal. These are the last 3 data sets that were most recently published using the search criteria that I looked at. But we've got some single cell RNA sequencing data from the human dorsal root ganglia, so that's in the spinal cord. We also have neural recordings of spontaneous metastasizing melanomas, so telling the difference between a melanoma tumor with high or low metastatic potential, which is a really exciting concept to think about, I think. And then cyto and chemo architecture of the rat spinal cord. So just in these 3 areas, we've got 3 different species. We've got 3 different modalities. We're looking at 3 different areas of the body. But what they all have in common is that they're all related to the peripheral nervous system. They all are processed and presented in a similar fashion. And that's something that the Spark program does, so that as data sets come in, regardless of how they were created, what they were looking at, or who performed the research, we provide a systematic method for how the data is presented to you, so that you can start to get used to how the data is available and start to expand your view. So on the Spark portal, we provide a faceted search. And so it's easy to find what you're after. You can select by organ system, you can select by species, and really dial down with categories, or searching within modalities in order to pull up different data. This is growing every day. And we're super excited to see how it continues to expand. These data sets are not all about the autonomic nervous system either. As you saw from the previous slide, we had the spinal cord, metastatic potential of melanoma from the skin, so skin tissue, and then the dorsal root ganglion, so sensory information about the spinal cord, all brought together from different funding mechanisms, and all keen to just bring their data to the same location. So Spark's mission is to bridge the body and the brain by bringing together diverse scientific data to increase the visibility and discovery through open science. The Spark portal is an open neuroscience and systems physiology platform containing multi-species data, knowledge, computational modeling, and spatial mapping. Individual researchers and research consortia are encouraged to become part of the Spark community. Exploration is easy with Spark's robust keyword search, which are extracted from the metadata provided with each data set. Visual and contextual search is provided using dynamic multi-species maps. These maps are annotated using a powerful knowledge base that provides connectivity information gleaned from expert input, published literature, and the contributed data sets on the Spark portal. The connectivity knowledge base is continuously growing and will soon include male and female reproductive system circuitry, as well as sensory and motor neuron populations. This fall, new tools will permit the scientific public to share feedback, citations, and annotations to enrich the map, providing even more open science opportunities to the portal. So far, the Community Hub for Peripheral Nervous System Neuromodulation Research has over 300 data sets and models from over 500 contributors. We're building partnerships across disciplines and consortia, which increases the diversity of data and the opportunity for cross-pollination. So Spark offers a scalable and sustainable platform for curation, management, archival, and data sharing. We organize and present raw and primary data, protocols, code, open hardware platform, and designs. And in order to make that accessible for everybody, we provide dedicated customer support and software tools to help the researchers contribute their data sets and methods. The Spark Data and Resource Center creates maps in the knowledge base for exploring this data and the PNS connectivity. We also create tools and 3D organ scaffolds for mapping data spatially and for performing computational modeling using our web-based simulation platform. However, we all want to look at data in a greater context, and meaningful sharing of scientific data requires an integrative approach beyond just ensuring that files are publicly available. You need to know how the data sets were actually created in order to leverage them as a jumping board to do something new with them. And so in order to do that, the data sets need to be well documented, they need to be standardized and semantically annotated. Importantly, we feel that they also need to include detailed experimental protocols and usage notes and any tools that they use to create the data. So it's this entire ecosystem that represents what these researchers actually did in the course of doing their experiment that allows others to sort of engage and understand how they can conceptualize using the data as well. So we provide, as part of the process, a team of professional curators that are ready to assist during regular office hours to help researchers adhere to these data standards. This provides a cohesive presentation that establishes the keys to those rich exploration potential by others. So each Spark dataset includes structured metadata files, a predictable folder structure, and explanatory files, regardless of the type of data that the dataset actually includes. It could be code, it could be a simulation model, or it could be a wet lab experiment where you're looking at physiological responses and neuronal tract tracing. All of those will all have the same overall structure so that adhering to standards means that you know what to expect when you walk into a data set. We also fully support the credit and attribution for the research groups who contribute their data. All contributors to the data sets are listed as authors, and we provide a full citation, including a DOI, for each data set. And we conform to the best practices for making data count. Looking at the data set number along the lower bottom of the screen here, you can see on this plot on the left that data sets on the platform are broadly accessed, regardless of what type of data they actually are. There's one in particular that's been really heavily accessed that we wanted to just call out for a bit, because we've learned that this particular data set is actually used in teaching an undergraduate course. So you can begin to see how sharing your data openly can open opportunities for expanding the context for others to grow and use your expertise. Likewise, protocols are very popular, which further underscores just how useful these data products are. It's not just the interpretation in a scientific paper that is useful. It's how you came to instantiate that experiment that's really useful to other scientists. And so open science really enables that to happen. The reuse of Spark data is something that we're super excited about. Like Ben mentioned, we're seeing growing evidence of dataset reuse as well. For example, these two papers on the left utilize datasets identified there on the right that other scientists contributed to the Spark portal. It's very exciting to see this growth, and we expect as more and more people become familiar with the process of sharing their data in an open way, they begin to see how they can also use other people's data to expand their perspective. This isn't our only view of open science, however. Since 2020, we've hosted a global virtual codeathon to bring together data enthusiasts to get creative with the Spark Data and Resource Centers platforms, as well as the data on the portal. It's a ton of fun, and a number of participants have returned year over year. Please consider joining us next summer. Lastly, Spark embraces the mission of open science by amplifying the scientific effort within the community. Community members are encouraged to tell their data story. So not just the interpretation and importance of their experimental publication, but the story of how the data is collected and why. these are Spark stories, and they provide ways for scientists to interact with other scientists and make just science more accessible in general. It's really exciting to see this creative side of science. And that's one of the really fabulous things about open science from my perspective. So thanks for your time. And if you have any questions, you can check out the Spark portal at spark.science. And if you're attending the Society for Neuroscience conference, come see Ben and I and our teams at our respective booths. We're right next to each other, and we'd enjoy seeing you in person.
Well, thank you, Dr. Tappan. The Spark portal is such a useful and accessible resource, and fascinating, compelling, publicly relevant research as well. You know, at Noggin, we work with many K-12 students who are undergoing, as young people and parents and teachers know very well, you know, tremendous change in their ability to regulate their emotions, which is directly relevant to the vagal autonomic activity and how that changes across childhood and adolescent development. So I'm having very positive autonomic reactions myself. Increased heart rate, for example, that reflects the excitement that I am feeling about greater access to federally funded research information, you know, that has such great relevance to us all. So I'd now like to open it up to questions of our experts. And as a reminder, please provide any questions you have in the Q&A box. There are a number of questions here. I just wanted to get to at least one or two. I know that there was a very interesting question here about how do you manage ethical considerations around data privacy in open science, especially when dealing with sensitive clinical data? And that's to any of you.
Spark. So for sharing data with Spark, it is important that you share de-identified data. So the requirements of the data sharing is made for the research teams themselves to support and manage. And that requires that they share de-identified data and obtain informed consent from any participant that they are working with as a part of their research paradigm. So that's how we handle those ethical concerns. Ben, is there anything you'd like to share?
Yeah, I mean, basically the same for us. We require any data from people to be de-identified before it's submitted.
And another question is, Ben, you mentioned NIH requiring their grantees to share their data. What happens if they don't comply?
Has yet to be seen. You know, that is a big question, right? If you make these rules, how do you enforce them? Our role is really to make it as easy as possible to comply with these rules, and kind of leave it to the NIH to figure out what to do if people don't comply. So far, we've seen people really excited about having these tools and being able to share data well. I'm sure there will be others that maybe there will be others that are not as excited to share data. So yeah, we'll leave it to them.
So just so everybody knows, the questions that were asked in the Q&A will be answered. Our speakers have agreed to, answer them offline. So we're at the end of our hour, and I want to thank our speakers, Drs. Dichter and Tappan, for your insightful explanations and overview of the important work that you're doing to advance the importance of open science. For more information on ABC, Neurodata Without Borders, or Spark, or Northwest Noggin, please visit our websites. Thank you very much and have a great day.